Meiosis by Hunter Atkins, Madeline Mitchell, and Abdul Sadiq. As we move along, you will notice that there are several highlighted key terms throughout. It is important to understand these key terms for that they will give you a better understanding of the meiosis process. What is meiosis? Meiosis consists of two stages known as meiosis 1 and meiosis 2, and they are responsible for the production of gametes. Gametes are known as mature haploid, male or female germ cells which are able to unite with one another of the opposite sex in sexual reproduction in order to form a zygote. To further add on to the definition of meiosis, it is known as the process of cell division in which gametes are made. In the process, it begins with a cell that has double the normal amount of DNA, and it ends up with 4 non-identical haploid daughter gametes, and this occurs after 2 divisions, one in meiosis 1 and one in meiosis 2. The first division occurs after metaphase 1 and is known as the reduction division. The second division occurs after metaphase 2 and is known as the equational division. Interphase. Similar to mitosis, meiosis is preceded by an interphase that includes 3 stages. These stages are known as G1, S, and G2. During interphase, the chromosomes are relaxed and indistinguishable as diffuse chromatin. After interphase, prophase 1 begins. Prophase 1 is a rather lengthy stage, and it is divided into 5 substages. These 5 substages are known as leptotene, zygotene, pachytene, diplotene, and diakinesis. Leptotene is the first of the 5 substages. During this substage, the chromosomes begin to condense and become visible. Zygotene is the second substage. In this substage, the chromosomes continue to condense as they did in the leptotene substage. However, now homologous chromosomes pair up and begin synapsis. Synapsis can be defined as the close pairing of homologous chromosomes. Each homologous pair of synapse chromosomes are known as a bivalent or tetrad, consisting of 4 chromatids. The next substage is known as pachytene, and in this substage, the chromosomes become shorter and thicker, and a 3-part synaptimal complex develops between homologous chromosomes. Diplotene is the 4th substage, and this is where the centromeres of the paired chromosomes separate except at the chiasmata. Each location where 2 chromosomes cross is called a chiasma, and multiple of these are known as a chiasmata. Lastly, the final substage is known as diakinesis. This is when chromosome condensation is continued and the nuclear membrane begins to break down and degenerate and the spindle forms. This now sets the stage completely for metaphase 1. Before continuing to metaphase 1, it is important to understand another key term, crossing over. Crossing over can be defined as homologous chromosomes that exchange genetic information, and this takes place in prophase 1. This is essential because crossing over generates genetic variation, and this is necessary for the proper alignment and separation of homologous chromosomes.
Following the completion of prophase 1 comes the next step of meiosis, metaphase 1. As we enter metaphase 1, the homologous pairs of chromosomes align at the metaphesial plate, forming a tetrad. A tetrad can be defined as a close association of 4 sister chromatids of 2 homologous pairs. This is a step of meiosis where Mendel's law of independent assortment can be applied, as these homologous pairs align randomly, allowing alleles to separate into gametes independently. For example, if we had 3 pairs of homologous chromosomes, aka 3 tetrads, there are 4 possible ways for the 3 pairs to align in metaphase 1. After this random alignment occurs, and following the completion of metaphase 1, leads to the random distribution of these pairs into 2 nearly divided cells. This defines a process of anaphase 1, and this is where these randomly aligned homologous pairs will separate, being pulled by spindle fibers into opposite poles of the cell. This phase can be defined as a second mechanism to generating genetic variations in newly formed gametes. Following the conclusion of anaphase 1, approaches the stage of telophase 1. In this phase, the spindle fibers disintegrate and the nuclear membrane reforms around the recombinant chromosomes, forming 2 nuclei. As this is happening, cytokinesis comes into play. This is when the cytoplasm divides as it is cleaved, resulting in the division producing 2 haploid cells. This division can be defined as a reduction division. This concludes the processes involved in meiosis 1, leaving the cells in a limbo prior to the initiation of meiosis 2. This limbo or period of rest is called interkinesis, and in this phase, the period of rest is generally short-lived, eventually entering the phase of prophase 2.
In prophase 2, the nuclear envelope dissolves in each cell just like in prophase 1, and the diploid chromosomes start to condense into sister chromatids. Diploid means there are 2 complete sets of chromosomes paired together. Then, duplicate centrosomes will form and migrate to the opposite ends of the cell. In metaphase 2, the centrosomes finish migrating to the poles, the nuclear membrane completely dissolves, and the sister chromatids are lining along the midline. Microtubules, known as spindle fibers, are connected to the centromeres and attached to the chromosomes at their kinetochores. These spindle fibers will pull apart the sister chromatids. In anaphase 2, this is where the sister chromatids are being pulled apart at the kinetochore by the spindle fibers and become 2 daughter chromosomes. In telophase 2, cytokinesis is beginning and the 2 daughter cells start to turn into 4 gamete cells. Gamete cells are reproductive cells and the goal for meiosis 2. The chromosomes are unraveling into chromatids and the nuclear envelope is starting to form again, as well as spindle fibers are dissolving. In telophase 2 and cytokinesis, the ploidy level is 1, which means they are haploid cells. The ploidy level is how many chromosomes a cell has. Finally, in cytokinesis, the daughter cells complete splitting and become 4 haploid gametes.